First, I want to say how nice it is to look out and see faces in real world, not on Zoom. This has been a great conference already so far, so it's been great to have this. We're based in New Orleans, so it's fun to have this in our backyard, and certainly want to say thank you to NCATS for putting this on and Hopkins and CAT for all the work that they've done setting it up. What I'm going to be talking about today is a case study in chemotherapy-induced peripheral neuropathy using one of our platforms, which we call a nerve on a chip. First, about the company and our team. We were founded in 2014. We've licensed our platforms from Tulane University and Johns Hopkins. I'll talk a little bit more about that. And then we're a growing team of 27. I'll say in the front, if anybody's looking for a job, we're currently hiring right now. So check out our website or catch one of our team after this. The two platforms that we utilize, I'll primarily be talking about the NerveSim model today, which is our nerve on a chip. That's used for neurotoxicity. Obviously, the case study I'll be sharing, but increasingly we're moving into the drug discovery side. I think the entire field sees that the impact there is potentially larger, and really, we want to capture that. And I think that's where the impact is. Obviously, Hesperos has now put in data into an IND filing. The reality of this is starting to show up. We also, towards that end, can work in rare diseases, ALS being one of them. The other platform is our BrainSim platform. That is a CNS model. And you saw, or if you were in the session, Johns Hopkins is where we licensed that, and they presented some data on oligodendrocyte precursor populations and ultimately myelination in that system. So it's a really elegant system. Background on peripheral nervous system, not something everybody here is maybe familiar with. It's basically all of the nerves outside of the brain and spinal cord. And these are really important, but frankly, understudied. When you look at, basically how this functions, you have the dorsal root ganglia and you have motor neurons that synapse either onto the skin, skeletal muscle, or the enteric nervous system. And we're going to be focusing today on the dorsal root ganglia as that interface between the sensory nervous system and the spinal cord. This is implicated in a big way in chemotherapy-induced peripheral neuropathy. Unfortunately, over 2/3 of patients who undergo chemotherapeutic treatments end up with a peripheral neuropathy. And this is not just a short-term problem. A little under 1/3 of them still have these issues after 6 months. And these are big issues. They affect mobility, chronic pain. And you can see, because these are such a big deal, they actually change the way patients are treated. As you see the numbers here, it can lead to kind of decreasing the potential efficacy by reducing the dose, and even some patients just stop altogether. And this has become a bigger focus within the pharmaceutical industry recently. And what we went to do here in our qualification study, basically these chemotherapy neurotoxicities kind of manifest in a couple of different ways. And we chose 4 of the primary classes that acted at different mechanisms. One of those is platinum compounds, oxaliplatin in particular. You can see where they are affected here. I'll kind of go through how we demonstrated that, so I won't spend too much time here. Bortezomib, a little bit of overlap there. Of course, there's overlap with all of them. Then we picked the taxanes to start looking kind of at the other 2 as well, and vincristine to cover the spectrum of these toxic mechanisms. Just background on the NerveSim itself. We took a tissue engineering approach to build a 3-dimensional scaffolding system. I'll show you a really nice video to help you understand what that looks like. But if you look at the image in the top right, in green there, we have beta-3 tubulin, and you can see we have this really nice polarized neural fiber tract. And having this 3D interaction between Schwann cells and sensory neurons, motor neurons, whichever the case may be for a given application, we get this really nice 3D compact myelination that you can see in the EM images below that. The other thing that we're able to see here, another clinically relevant metric, is population level electrophysiology. So we're not looking at the single cell level. We're really looking at a summation of all of the tens of thousands of neurons, axons that we're recording from, and look at that compound action potential, and then most importantly, the conduction velocity. And this is really one of the primary metrics that a neurologist would look at. And we, as I've already described, right, we're able to kind of pick and choose the cell types that we want in, corresponding with the application that we're looking at. We take those in a very defined ratio and density, and we form them into spheroids to basically get the basis of those cell-to-cell interactions. Then we engineer that into the three-dimensional scaffolding system that is the nerve sim or nerve on a chip. We're able to isolate the neurons in this circular region of a keyhole and induce, constrain, define the both neurite outgrowth and glial migration, where we're getting tens to 50,000s of axons. We can come in after introducing a drug, look at what changes functionally, what happens to that conduction velocity, what happens to that compound action potential. And after we know functionally we see this decrease or this rescue, we can come in and we can look at the histology of this. This is analogous to a nerve biopsy. Again, clinically relevant, but then also in vivo relevant, which is really important for a lot of our pharma partners. What you're looking at here is toluidine blue staining, look at myelination, and then again, an image of electron microscopy looking at that really compact, thick myelination. The case study, we set out to multiplex our system so that we could start parsing out those toxic mechanisms that I talked about at the beginning. Already told you about the EPHIS, the histology. We're also able to, because this is set up in a relatively traditional in vitro layout, we are able to run more traditional studies like cell viability, looking at mitochondrial function. We can sample supernatant. So all of the things you might look at otherwise, we can stack on top of these unique metrics. Here, we're using the compounds that I showed you before from A to D. Those are the 4 chemotherapeutics. And then in E and F, those are the negative controls. And you can see, across the chemotherapeutics themselves, we see this dose-dependent response in the nerve conduction velocity, which is the black line that you see there, as well as the compound action potential that you see in gray. And what you can start to parse out here is some of them cause a much faster functional decrease. And I'll talk about some of the IC50s so that we can kind of understand, how to separate those out. We really wanted to understand not just seeing functional differences, but make sure that that didn't correlate directly to the cells dying, right? Those need to be separated for us really to look at the mechanism. And that's what we did here. In red, you're looking at a cell viability assay. And black is the same conduction velocity I showed you before. And with paclitaxel, you can see along the doses that we worked with there, you see that functional decrease. We do not see a decrease in the viability. That matches with the mechanism here. A microtubule stabilizer would be expected to act this way. When you look at oxaliplatin, highly cytotoxic, you see that those really correlate well with each other. That acts at the DNA level. Bortezomib, you see that they track each other, but arguably that function is a more sensitive metric. And we're really reinforcing that the mechanisms that have historically been seen are showing up here. And then finally, with the vincristine, another microtubule affecting drug, we see that viability does not change. And what's interesting, this obviously plays out in the IC50, but we saw a unique result basically with the kind of differentiating the conduction velocity and amplitude. You can see with the two, obviously there was no change in viability. We already kind of could see that pretty obviously. Bortezomib, you see preferentially this decrease in both functional metrics before you see the change in viability. Oxaliplatin, you see nerve conduction velocity itself really shows up as a more sensitive metric, in the amplitude and IC50 for the cell viability actually are exactly the same. We went next to look at mitochondrial function. This is a mitochondrial membrane potential dye. And what you can see, I think that's pretty obvious, FCCP, which is in F, is a positive control, basically kills off the mitochondrial membrane potential. But then with oxaliplatin and bortezomib in particular, you see a dose-dependent decrease there. And when we look at this quantitatively here, we're basically looking at pixel intensity. And you can see, FCCP obviously significant, oxaliplatin and bortezomib at the high concentrations are, but then oxaliplatin, you see a difference between the two doses as well. Went in now to look at myelin basic protein, what was happening at that level. In red is an MBP stain. In green, you're looking at beta-3 tubulin stained neurites. And what's pretty obvious from a qualitative perspective, at least, is with paclitaxel and vincristine, you see degradation of that MBP, where you don't in oxaliplatin or bortezomib necessarily. Again, tracks with those tubules being a primary mechanistic toxicity manifestation. We look and go in and are able to evaluate the EM images here. And this lets us really dive in, to the ultrastructure where we're able to look at fiber densities, whether it's myelinated or not, the thickness of the myelin, where we might see some myelin degradations. Can even, as you see in E, look at the nuclei themselves, see what's going on there, and then looking at fiber diameter. And that's certainly an important one. And when we put all of this structural data together, all of this histology data together, you see some interesting trends that show up. Paclitaxel, for instance, we saw that MBP staining, but then we see fiber diameter is affected in a big way. Microtubule stabilizer, so you would expect that those diameters would see that tubulin build up and sort of expand when they're not in a healthy state. Oxaliplatin here, the eccentric nuclei that tracks immediately with the DNA damage. And in bortezomib, we see some of that, but we also see that the non-myelinated fibers in particular are affected. And then with vincristine, this is really happening with the density. So those fibers are degrading to microtubule inhibitor. So you would expect that breakdown to be happening. And you can see, at the last box at the bottom right, that those fibers are degenerating. We're able to now, the electron microscopy is a very slow way of getting back these responses. So we've been able to refine some of our metrics, and we're really working a lot more with cryosectioning. You're able to, with this system, manipulate it in a very big way, specific way. And what you're looking at here is basically a cross section where we're able to slice this up. In the top left, you're looking at a cross section right where the spheroid is. So we're able to look at those cell bodies. And you can see that up close on the image in the right. We're able to then section farther and farther down the channel to see what's happening, more proximally to the cell bodies, what's happening more distally, really continue to differentiate between axonopathy and neuronopathy and understand, not just obviously that a drug is toxic, but why. And that's really important when a pharmaceutical company is trying to improve their compound, maybe tweak it, whatever it may be. They really need to understand in sort of a simplified MPS model what's going on. And antibody drug conjugates are a big one that we look at. They're I think, something that's been proven very effective, and now they want to make them safer. So it's been really fun learning from our pharmaceutical partners on this, really how to tease out whatever data that is they might be needing. What you saw along the way here, we recapitulated the mechanistic toxicity for these four major classes. The high content data, once you put it all together, really lets you parse out the nuances of this. And where we're moving now, as I alluded to before, is really neuroprotection. So we're working on a big case study here demonstrating that we can recapitulate data that's been published, where you're looking at, whether it's co-administration or administration of a neuroprotective drug after the damage, seeing that reversal or seeing that protection. And I'm going to kind of close by showing you a little bit of a teaser for a poster that we have later today. We have launched a new platform upgrade to improve our throughput, which is a really big step. Because this system is unique in its geometry and the three-dimensional nature of this, we had to basically take a step back and completely customize the electrode layout, the geometry, the materials we were using to make sure it interfaces with the 3D environment. And as you can imagine, that's not trivial. 3D provides a lot of advantages, but it also makes a lot of things difficult, right? Imaging, being able to interface with the system. And what's nice about this, not only does it increase the throughput as well as the reliability, it allows us to gather more metrics from the system, and it lets us test the system with time as well as across multiple electrodes. This is an example of the layout where we have 10 electrodes through, essentially that same keyhole shape. You see we have the circles on both ends just for a little bit more flexibility. And we're able to grow the axons directly over these electrodes. And we have that in our 24 well system. So we're increasing the throughput, certainly, not yet at 96 well, but we really position this as high content, more so necessarily than 96 well, MEA, something like that. When we start to kind of look into what this actually looks like, I think I mentioned, or if I didn't, I'll say now we're able to stimulate and record from each of the electrodes that you saw before. This is an example where we stimulated at electrode 5. So obviously distally, down the neurite outgrowth microfiber tract. And then we're able to look at that signal that propagates both proximally and distally. And you start to see some signals pop up as we move closer to the cell body. And as more fibers presumably are recruited, we see that these responses are larger in amplitude. And then ultimately, when we see it at the cell body, we see these really big somal responses. And we're able to gain something out of each different location, looking at distance between between the stimulus and multiple of the electrodes all the way back to the cell body. And within each of those are different metrics. And those endpoints are basically drawn out of the data that we analyze with velocity distribution. You see here those 3 distinct peaks, which I'll show you correspond to a really important biological metric. And we're also able to, and this is a really unique one that's opening up some doors that we're very excited about. What you're seeing on the right is basically from top to bottom, increasing the stimulation. So increasing the current of that stimulus and looking at the response. And you can see, at the lowest current at the top, we don't see any response. But then what you see is, that response begins to show up with the increasing current. And then you see more of those responses at the highest level there. And that's really important if we're talking about using this as a pain model. We can see sensitization. We've done a preliminary study here where we knocked out the signals. And then when the signals came back, not only did we demonstrate that they were biological, but they were actually more sensitive to this stimulation. And so we're about to embark on a project with a big pharma company to really dive into this pain application. Neuropathic pain is a big problem. I'm not going to run through all this. I encourage you to go to poster number 31. Corey Roundtree, our lead scientist, will dive into this much more than I will. You can see here with all of those different metrics, and I didn't even talk about half of them on the left, it corresponds to those different histological endpoints that I referenced already. So we're able to glean out of this, basically what's happening with the structure without even necessarily having to go in and look at that structure. Now, of course, we need to demonstrate that correlation many, many times, but the imaging is typically one of the biggest bottlenecks. So we're really excited to package this all into one set of endpoints. Again, go check out that poster 31. This is a really quick demonstration of a little bit of the data. So the pink is not showing up, but basically the entirety of that circle that you see there is the vehicle control. And you can see around the outside, the different metrics. And I'm not sure what's going on here, but the colors are mixed up here. So I'm sorry. The green is, the lowest dose of paclitaxel that we tested here. That's about a medium strength of toxicity. And then blue is increasing that. And as the circles shrink or those points shrink, that means that those values are going down. And what you can see with amplitude of peaks on the left, velocity of peaks, there's a dose dependent response there. And what's interesting on the right, when you look at medium, slow, and long distances, we see that basically immediately with that medium dose, the medium and long distances are knocked out. But we see specifically that these slow fibers are decreased in a dose-dependent way. And that's really important to be able to differentiate between those different fiber types, between those different populations. And so, with that, I would like to take any questions, and please go see poster 31.
Okay. We have time for a couple questions. Please step up to the mics. So I have a question. When you're looking at the different fiber types, do you think that you're actually recruiting as if you would during increased activity, that you have your dominant fibers and then you had your less dominant fibers and you recruit them as you increase your stimulation activity?
That's what we think, definitely. And we're really going out to demonstrate that more and more as we look at that excitability, the dive more into the different stimulation levels. And then we really want to prove that with some different compounds that are known to maybe knock out specific populations and see, when that begins to manifest itself. So we do think that, but we want to validate that further just to make sure that that is what we're seeing, because obviously this system is an artificial microfiber tract, right? And so before we state that too strongly, we really want to be able to parse that out from a number of different angles. But if that bears out, that's a really big deal that we're excited about.
No, I agree with you. Any questions? All right. No more questions. Larry, thank you very much.
Thank you very much.